from the laboratory of Guam, and Alex Lee is going to be talking about viral gene delivery.
Good morning, everyone. My talk today will be talking a little about back to specific cell type with engineering single-chain antibodies. One of the reasons why we're interested in gene therapy is that gene therapy is the introduction of a functional gene into target cells to provide therapeutic advantages. So why are we interested in targeted gene delivery is as follows. In our body, there's a variety of different cell types. There are some good cells and some bad cells, for example, some tumor cells. So we want to be able to deliver our therapeutic genes into only the tumor cells that has a characteristic that can be recognized by our engineering viral vector. In our system, the cells type of study is CD20 positive cells. CD20 positive cells is restricted to only B cells, and it's not expressed on either precursor lymphoid cells or the vast majority of plasma cells. And an important thing is over 90% of the non-Hodgkin lymphomas are CD20 positive cells. That's why we picked this cell line as our study model. So here is a brief diagram of how we synthesize our recombinant lentivirus. First, we'll introduce 4 different constructs into the nucleus of our virus producing cells. One of the constructs includes a GFP reporter gene flanked by the 2 LTRs. This GFP reporter gene can help us to study whether the toxin transduction occurred or not. We also introduced the viral gap and PO, which the gap protein is essentially supply the viral structure protein, and the PO is used for the viral enzyme. And we have also included 2 other components in our system, which include an antibody. An antibody in our system will be used to guide our recombinant lentivirus to target only the CD20 positive cells. And then we also have to introduce a feature chain to help the virus to be released, to release its genome information once upon the successful infection. So once all 4 of the constructs are present inside the nucleus cells, the protein will be synthesized as well as the viral RNA. And once they get together around the cellular membrane, the lentivirus will naturally bud out. And then when they bud out, it will carry a piece of the cellular membrane, and along with the proteins that are associated on the on the cellular membrane. And finally, we'll have a functional lentivirus that carry both the antibody as well as a future genetic molecule. So why are we interested in studying single-chain antibody to display on our viral surface as well? In order for the natural antibody to form, we need to have 4 different constructs to be synthesized together. One construct has to synthesize the constant region of the antibody. One has to synthesize the variable region. And also, we also need 2 associated proteins to anchor the antibody, which includes a human IgG alpha protein as well as a human IgG beta protein. So being an engineer, we think maybe if we can simplify the production of an antibody, maybe we can increase our viral transduction. So what we have come about is utilizing a single-chain antibody, which we use a GS linker to fuse the variable light chain to a variable heavy chain, as well as an introduction of a transmembrane domain. So in this regard, we also need one construct version that will display both the variable region of variable light chain, variable heavy chain, as well as our single-chain antibody. So an experimental outline for this experiment is we'll synthesize 8 different targeting viruses. 4 of them are coming from one version of a single-chain antibody paired with a 4 different version of our previously established future genetic molecule. as well as a different version of the single-strand antibody paired with the same 4 different version of the future genetic molecule. For control virus, we will include a B12 antibody, which is known to not interact with the CD20 antigen, and it should not be able to bind or transduce. But we also pair this with a future genetic molecule. And we also include 2 different kinds of antibody-only virus without a future genetic molecule, and a PHVG pseudotype lentivirus that is known to be able to infect a variety of different cells at high titer. We'll use 2 different cell lines as our target cells. One will be a 293 CD20 cells. Essentially, we engineering the cells to overexpress CD20 antigen. And we also include a parental cells as our control cells. Then we have did an in vitro study of our target transduction by incubating our targeting virus with either the parental 293 cells, the control cells, or the target cell, 293 CD20 cells. So as you can see here, we can observe when the targeting virus was incubating with the targeting cells, we see a 30%, roughly 25 to 30% of the cells were infected. Whereas when we incubating the same virus with the control cells, the CD20 negative 293 cells, we only see about roughly 5% backbone infection was observed. As well as when we incubate only the antibody-only virus, almost no transduction was occurred. As well as when we incubating our B12 control virus with either the targeting cells or the control cells, no transduction was observed. Then we give a proposed mechanism of how our system will work. So first, our system will, our recombinant antivirus will bind to the antigen on the cellular membrane via the antibody-antigen interaction. And then this interaction will actually trigger an endocytosis event, which will bring the lentivirus into the endosomal compartment. And inside the endosomal compartment, the pH will gradually decrease, and that decrease will actually trigger the conformation change for the future genetic molecule. And the future genetic molecule will then open up both the viral membrane and the cellular membrane, and our capsid will then be released. Then we perform a cell virus binding assay to show that the first step is indeed occurred via the antibody-antigen interaction. So what we have done is we have generated a targeting lentivirus, and then we incubate with either the 293 cells or the 293 CD20 cells at 4 degrees for up to an hour, and then followed by a P24 staining. The P24 here is the viral capsid. So as you can see here, in the 293 cells, the CD20 negative cells, no signals were detected on the cellular membrane. Whereas when we incubate our targeting virus to the target cells, the 293 positive cells, we see a lot of signals start to appear on the cellular membrane. So the second step for our viral gene delivery is the pH-dependent fusion activity. So in order to test that hypothesis, we perform a neutralization assay in which we incubate our targeting virus with the target cells, but also with a different concentration of the bifidobacterium drug. The bifidobacterium drug is known to neutralize the pH environment inside the endosome. So by increasing the amount of bifidobacterium drug, we'll see an increase of the pH environment inside the endosome, which will lead to a decrease of our viral transduction. And as you can see here, it's indeed the case. By increasing the amount of bifidobacterium drug, we do see a decrease in our target transduction. So in summary, we have developed a targeting lentiviral to specific cell types. And our targeting lentiviral has to have 2 components: an antibody that's used to recognize the specific antigen on the cellular surface, and also a fusion genetic molecule that's used to help to release the viral core into the cells. And we have also further engineered a single-strand antibody to be displayed on the lentiviral surface to transduce the specific cell types. And I would like to thank my advisor, Dr. Tim Wen, and all the lab mates for their support.
questions?